All right, so here we're going to talk about regulation of fatty acid oxidation, as well as the production and regulation of ketone bodies. And so we'll talk again about beta oxidation, but how it is either turned on or turned off. And then we're going to describe the synthesis of ketone bodies, which is from acetyl-CoA, which of course during beta oxidation of fatty acids, you make a lot of acetyl-CoA. And then we're going to talk about regulation of ketone bodies. Of course, we're here in the liver because the liver is where you're making your ketone bodies. Now, oxidation of fatty acid is going to have control over needs, of course. So when you're in the fed state, insulin, you're going to turn off this process. And when you're in the fasting stage, you're going to turn on this process. And so glucose levels is really going to regulate your insulin glucagon levels, which then will regulate whether or not you're going to be making fat or breaking down fat. So insulin release is going to be during the fed state, as we know, when you have high glucose, so you don't need to break down fatty acids. Glucagon levels in fasting is when you have lower glucose levels. So now you do need to start making glucose, for example, in your liver. So you don't want to use glucose for a fuel. You want to release it, and therefore you have to rely on alternative sources for fuel. And one of the main ones is fatty acids. So when energy is needed, you're going to favor beta oxidation, and when energy is not needed, you're going to favor fatty acid synthesis, which makes sense. All right, so regulation, you're going to, of course, have hormones. Hormones are going to control whether you're going to activate your kinases or phosphatases. They're going to be sensitive to your energy demands. So hormones are going to be critical for releasing fatty acids from your adipose tissue so that they can enter the blood. So hormones will lead to an increase of fatty acids. Now, the other thing that's going to regulate beta oxidation is, of course, going to be your pool of acetyl-CoA, or not CoA, sorry, coenzyme A. So remember, in our TCA cycle, or we're here in our TCA cycle in our mitochondria, you're going to generate CoA pool by converting acetyl-CoA with, or combining acetyl-CoA with oxaloacetate to make citrate. So that's going to release that CoA, and that's going to fill that CoA pool so that you have CoA present so you can use it to generate your activated fat in your mitochondria matrix. So you also have a CoA pool in the cytosol, and that is going to come from other processes in the cytosol that release your CoA. But CoA present is going to be required to activate your fatty acid in the cytosol, and then again activate your fatty acid in your mitochondria matrix. Now, another thing that's important is malonyl-CoA. So malonyl-CoA is going to be part of the process of making fats. So when you have your insulin, you're going to start to make fat. And so this is going to take acetyl-CoA through acetyl-CoA carboxylase and make malonyl-CoA. So acetyl-CoA is a 2-carbon, malonyl-CoA is a 3-carbon. So you're adding a carbon through the carboxylase, and this is turned on to make fat. Malonyl-CoA is going to shut down beta oxidation by preventing the transfer of your fatty acyl-CoA to the mitochondria matrix. So your malonyl-CoA will block your carnitine CPT1. Let me do this. So it's going to shut down that. So this is going to turn off oxidation and turn on fatty acid synthesis. So synthesis and oxidation do not occur at the same time. One is going to override the other, depending on the needs of the cell again. And it all goes back to ADP, ATP. So ADP, ATP, of course, is going to be important. That's going to tap into our pool of cofactors, whether they're available or not available. So remember, in order to make, or in order to do beta oxidation, you need a pool of NAD plus, and you need a pool of FAD. If they're reduced, then you're going to turn off beta oxidation of your fatty acids. Okay, so now let's move into talking about ketone bodies. So we're in the fasting stage, we're in the liver, fatty acids are being oxidized in order to generate energy. We know we need a lot of energy to generate glucose, and so your liver is going to use acetyl-CoA to make that ATP to make glucose. But your liver can do other things as well. So your liver will pick up fats, undergo beta oxidation, and generate acetyl-CoA. Acetyl-CoA can enter the TCA cycle, or it can become acetoacetate, and it can become what we know as our ketone bodies. So acetoacetate is going to be directly derived from acetyl-CoA. There is a second ketone body, this beta-hydroxybutyrate, which is going to be generated through a dehydrogenase that requires NAD, requires NADH. And then that's going to become from acetoacetate to beta-hydroxybutyrate. And this can go back and forth. So again, it's going to depend on the state of your NAD, NADH pools, for example. Now, ketone bodies are going to be released by your liver. Your liver cannot metabolize ketone bodies. And why? It has no enzyme for it. So much like when the liver has to release glucose, we're going to take our glucokinase and send it off into the nucleus. Well, in the case of ketone bodies, through epigenetics, the liver doesn't even express the enzymes to metabolize them. They make them and they release them. Your muscle cells loves to pick up your ketone bodies. Your ketone bodies can very quickly be broken down to acetyl-CoA, and then they can be used to make energy in your muscle cells. So synthesis of the ketone bodies is occurring in the mitochondria matrix. That's where beta oxidation is occurring. That's where you're making your acetyl-CoAs. And so from acetyl-CoA buildup in your mitochondria matrix, you're going to get a thiolase reaction, and this is going to generate, with two acetyl-CoAs, you can generate acetoacetyl-CoA. Now, this is only going to happen when you have high levels of acetyl-CoA, which again, what we saw with beta oxidation of your long-chain fatty acids, you make a lot of acetyl-CoA. So basically, in your mitochondria matrix, you're going to have acetoacetyl-CoA, and through a synthase, a 3-hydroxy-3-methylglutaryl-CoA synthase, you're going to generate your 3-hydroxy-3-methylglutaryl-CoA, and then you're going to have a lyase, which is going to generate acetoacetate, which is our ketone body. Now, acetoacetate can be released and enter the blood directly, or it can be reduced to generate beta-hydroxybutyrate. So here we have our acetoacetate, which is a ketone body. We have our beta-hydroxybutyrate, which is a ketone body. And we have our acetone, which is also considered a ketone body, but you can't use acetone. So acetone will get exhaled, and you lose potential in acetone. It's important to understand this is spontaneous. So when you are in a ketosis state in which you're a type 1 diabetic, for example, and you're not making enough insulin, so your liver doesn't know it's in the fed state, your liver is going to continue to oxidize fats. It's going to continue to make acetyl-CoA to high levels. It's going to also, there's going to be plenty of glucose around, so what's going to happen is your muscle cells are not going to pick up your ketone bodies as well. And you're basically going to increase ketone bodies. When you increase ketone bodies, you increase the spontaneity of the formation of acetone. And that's why people who are type 1 diabetics, their breath smells like acetone because they're exhaling the acetone. So also, under normal conditions, the ratio of the two ketone bodies that our cells can use is typically going to be 1:1. However, if you have an increase of ketone bodies, you want to push your keto body toward your beta hydroxybutyrate, because beta hydroxybutyrate cannot become acetone. Acetoacetate is the one that becomes acetone. All right, so now we're going to oxidize our ketone bodies. So again, we go back and forth via dehydrogenase between beta hydroxybutyrate and acetoacetate. Your oxidation is primarily going to occur in most tissues, especially muscles. They love them. Liver and red blood cells don't have the enzymes to metabolize these, so the liver never picks them up. The brain can metabolize them as well, and it's going to be very important during the fasting stage for the brain. Ketone bodies are going to move through the blood, and then you're going to have in the mitochondria matrix both acetoacetate and beta hydroxybutyrate oxidized, but beta hydroxybutyrate must be first converted to acetoacetate. And this is again by that same enzyme, beta hydroxybutyrate dehydrogenase. Okay, so once you have acetoacetate, you are going to, once you have acetoacetate, you're going to have activated succinyl. So you have succinyl-CoA, coenzyme A is attached to it, it's activated. And then you have a transferase to acetoacetate. So now you have acetoacetyl-CoA. So now you have an activated ketone body. And then you're going to have a thiolase. And this thiolase is going to generate 2 acetyl-CoAs. Remember, you made acetoacetate with 2 acetyl-CoAs. And you get 2 acetyl-CoAs back out of it. Beta hydroxybutyrate can go through the same pathway. However, it needs to first go through that dehydrogenase. All right, so homostasis. So of course, during the fasting stage, this is where we have our release of our fatty acids, and this is where we have our generation of ketone bodies by your liver. So the fatty acids are going to be released, picked up by your liver, other cells as well, obviously. We're just focusing on the liver. And the liver is going to generate a lot of acetyl-CoA. The acetyl-CoA is going to generate a lot of ATP. That ATP is going to be used to maintain blood glucose levels through gluconeogenesis. At the same time, though, you can make ketone bodies with that increase of your acetyl-CoA. And so you're going to have your change in hormones that are going to trigger your adipose tissue to split up the tag into 3 free fatty acids. And then you're going to have changes over time based on the number of fasting days. So you can see here that your free fatty acids and your acetoacetate don't change very much through prolonged fasting. However, what really peaks up here is your beta hydroxybutyrate. Your beta hydroxybutyrate is going to be favored because, again, it won't become acetone. Glucose levels will be kept relatively stable because of gluconeogenesis. So if you have enough fatty acids, you can keep your liver going. Liver can maintain blood glucose levels. It's going to kick out a lot of ketone bodies. It's going to favor beta hydroxybutyrate over acetoacetate. All right, so who uses ketone bodies? So again, skeletal muscle is going to love them. Muscle cells love them. They're going to be used by your brain cells, especially during fasting and for prolonged fasting. Now, in this case of starvation, of course, your skeletal muscles will switch to fatty acids and spare the ketone bodies for your brain. You're going to use ketone bodies in your adipocytes. Your ketone bodies can cross the placenta. And importantly, liver and blood never use them. All right, so regulation of ketone bodies. So again, this is in the fasting stage. So your fatty acids are going to be released from your adipocytes. You're going to have oxidation of fatty acids, so this is going to turn off synthesis of fatty acids. Your beta oxidation is going to give you a lot of ATP. You're also going to have a buildup of NADH. Your acetyl-CoA is going to build up because you have a lot of ATP, and so this is going to push towards ketone bodies and ketone genesis. So that's over here. All right, so in general, fatty acids are going to be used during fasting. Your fatty acid is going to be used by your liver cell, going to undergo beta oxidation, and this is going to be controlled, of course, by hormones as well as a need for energy. Your liver is going to convert fatty acids to ketone bodies, especially when there's an increase of acetyl-CoA, increase of ATP, increase of NADH. The ketone bodies include acetone, which is lost. So this is lost potential. Acetoacetate, a little arrow there, and D-beta hydroxybutyrate. Acetoacetate, beta hydroxybutyrate are going to be able to be interchanged by a dehydrogenase. So the beta hydroxybutyrate is reduced form. You're going to have acetoacetate that can be spontaneously form acetone, which you exhale and you lose that potential. Again, your ketone bodies cannot be metabolized by the liver or the red blood cells, but other cells in the body, pretty much all the other cells, can metabolize them. Your muscle cell loves them. Your brain will use them during prolonged fasting. During prolonged fasting, beta hydroxybutyrate is favored so that you don't lose your potential energy in the form of acetone. And of course, overproduction of ketone bodies when you have a lack of insulin, such as type 1 diabetics, can lead to acidosis or ketosis. And this is seen in type 1 diabetics and not type 2 diabetics.